And I know we're talking about, a little while ago, but fast forward to today, is it still the same? Biotech is the engine?
Well, it's still largely the case, but I want to talk a little bit more about the other modalities that we focused on in building new companies, because there are differences there. So this was what's called monoclonal antibodies. This development, really originated from the 1980s with Celtech as a pioneer and others seeing this as a way of making these more complex products that could then have a bigger impact on the biology of diseases. So large amount of capacity, mainly captive, but half the pipeline in small companies who then struggle to get to access capacity.
So there's a bottleneck.
And in 2010, I spoke to one of the leading consultants in our industry, and I told him what I was thinking of doing. And he said to me, "Why are you building capacity? There's plenty of it." And I said, "No, but it's mainly captive. Right. It's not accessible to these companies."
Right.
And then there was a second thing happening at that time, which was a new manufacturing platform was really coming to the fore. So back in the early 2000s, single-use technologies were developed. So single-use is where traditionally you would grow these bacteria within bioreactors, and the whole thing would be stainless. All the pipeline between the bioreactor and downstream processes, chromatography and filtration and other steps would all be stainless steel.
Right.
And it would all have cleaning systems associated with it. And for the bioreactor, steaming systems, so you could sterilize. Very complex engineering setups that took a long time to build, were expensive. And this new approach was to say, we're going to do away with a lot of that because we're going to use single use. So we're still going to have a stainless body, but we're going to line it with a bag. We're going to put in a very high quality plastic bag in which the bacteria can grow.
And the bioreactor, I guess just to put it simply, is like a piece of engineering that allows the little factories to do their thing, to create an environment that they're happy in. Is that right? And then you can pull off whatever element it is that it's producing, which will ultimately be used in the downstream drug. Something like that?
That's correct. And in some cases, which is something that Genzyme did a lot of, you can pull off continuously. And in other cases, you do it in a batch mode. But the principle's the same, that you're feeding with nutrients and the right temperature and oxygen or the lack of oxygen in order to create the right environment for the cells to grow. For these microbial in some cases or mammalian in other cases, so different types of cells.
Very interesting. And those cells differ because what the factories, them being the factories, produce differ too. It depends. That's
right. And it's really matching the right cell system, manufacturing system to the product that's been made. So for some more complex products, you need a mammalian system to manufacture those.
Very cool.
So single use then meant not just was the bioreactor lined with plastic, which at the end of the run, instead of flushing through with water and then chemicals and testing to show that you're down to parts per billion undetectable, instead you'd rinse out the bag and then you would dispose of it, which sounds like it might be wasteful, but actually studies were done to show that there's a lot less energy use if you do that versus this very extensive washing and cleaning and sterilization that's needed. But then from the bioreactor to the downstream units, all of that was disposable as well. So again, instead of having what literally would be like a Christmas tree of valves in order to steam and wash and set up the system for the next run.
Wow.
Instead, you remove a length of tubing, put another length of tubing in place that's sterile, and you're good to go.
One of the other benefits must be in pharmaceutical manufacturing, you have to prove that your cleaning is effective.
That's right.
So if you've got a single use, then that burden reduces.
Absolutely.
It's expensive and it's time consuming, isn't it?
Absolutely. And Genzyme's original standard was 2 weeks to turn around a bioreactor.
Wow.
Whereas with single use, you could literally turn it around in a day. Now, the paperwork probably means you need a few days, but that was no longer the bottleneck.
Wow.
So this new platform was just coming to the fore. A company in Massachusetts had recently installed 2,000 liter scale bioreactors. So this now was a scale that was meaningful in terms of the output that could be achieved. So we were able to use this new platform, and we built the first, what was branded a flex factory. So the first fully disposable 2,000 liter scale facility for clients in this site, this Johnson & Johnson site that we acquired in St. Louis. So
you must have raised the money to acquire the site.
So we eventually found the right investors.
You got the right investors. Yeah.
Plus, so this facility is in St. Louis, Missouri. And St. Louis is a great place to have a business, very stable. There are a few life sciences companies there, certainly not at the level of California or Massachusetts, but there's a hub there. Good university system, so graduates coming out with the right knowledge and experience to train up in these facilities. And a very supportive city and state that wanted to keep these jobs there and grow them. And so our main support was from an investor that we found, who was a great partner to work with, but then also really good local support as well.
Yeah. And it sounds like this factory was sort of totally state-of-the-art. One of the only ones with the new platform. Is that right?
That's correct. So there was an existing piece that was approved. So we made no changes to that because that would have been disruptive. So there was an existing piece to the facility, which we continued to run and produce product for Johnson & Johnson initially. And then we added on a clinical facility using this new state-of-the-art manufacturing capability.
And was the investor fully bought into you doing that? Because it sounds like new tech, maybe a bit risky.
They were bought in. They were great to work with. About 20 or 25% of their investments are in healthcare. But you know, this was a bit of a leap of faith for them, for sure,
and for you.
And for me, and of course, here was a site that had good site leadership and 160 people. So it was a really strong organization in place, but it wasn't a business.
Right.
It was a site.
It was a site.
So we had to add
mindset had
business
to change.
Development capability. We had to. There was a shift in mindset because it's different being part of a carve out, not really a startup, but a carve out new business versus being a hub on a giant international company's supply chain.
Yeah, and suddenly instead of meeting the demands of Johnson & Johnson as the sole customer, you bought it, set up the company, which was called?
So it's called Gallus Biopharmaceuticals.
Gallus Biopharmaceuticals, and there's a theme there that we'll touch on
indeed.
in a little bit. To you saying, well, actually, we want to service lots of different companies who might have tighter timelines than Johnson & Johnson. Because the small businesses, therapeutic companies, you know, now having invested in some and having worked with so many over the rest of your career as a CDMO entrepreneur and executive, that they have a fixed amount of cash, there's a burn, and they need to deliver, otherwise the companies might not survive. Right. So how did you go about changing the mindset of something that was stable? It's tough as the new leader.
Well, it's tough, but this was also an organization that had actually been through 5 other ownership changes since it was originally set up. So they were used to there being a change of the signs, but in some cases, not much else changing. So previously, Wyeth, another large pharmaceutical company, had owned the site. So there was a receptiveness. This wasn't something completely alien. What was different is exactly what you were saying, which is, hey, we're going to support smaller companies. We're going to do development work for them, process development work. Then we're going to scale their processes, and we're going to make clinical batches. And you're right, there's an intensity to that. There's nothing slack about our work with Johnson & Johnson. No. So We had an agreed production schedule, and a significant part of the organization was still focused on making commercial product, getting inspected by the FDA, and making sure everything stayed in good order.
Okay.
But what we did very early on was we got the organization together. We did this in groups, and we said, okay, what's working well today? What do we need to keep the exact same as it is? What do we need to change? And so we identified those things that would be different, being an independent, growing company, but no longer a satellite of a large organization. So
Not
we
where they were
identified
built in.
what exactly. Well, and they were part of it. It wasn't something done to them or given to them. They absolutely were part of identifying what were those changes that were needed, and how should we be organized, and what should we do differently, and what do we want to keep the exact same because it's working well and it's what's needed. Right. So that engagement was critical. I didn't have all the answers. It was the organization, the team that came up with the answers.
That makes sense. How did you build it, and what happened? What was the end outcome?
So we gave it a new name that nobody had heard of, and so here we were trying to offer services, and nobody knew who we were. So we went to conferences. The good news about working in our industry is that once somebody goes into the clinic, once you put a biologic into a human being, it's public information. So you can find out all the companies who are in clinical trials.
Okay.
And so we built a business development team that was able to identify potential customers. And they weren't just US customers, there were some European customers, and in fact some Asian customers as well. And so we would do outreach, try to find those who were looking. And then the way it works is sometimes a request for information is issued. So they want to know more about who you are and what your capabilities are.
Yep.
And then a request for proposal would be issued. And companies might be going out to 5 or even 10 companies asking for proposals.
Yeah.
So we would give them a proposal. And if they liked what we were offering, which really often was about technical capability, fit in terms of scale, access to what they needed, timing, quality for sure, which was a huge selling point we had because we were working from a basis of a team and an organization that had been doing commercial manufacturing for a number of years.
Yep.
So some of our competitors who had built a CDMO over time still weren't commercial. So that was a huge advantage we had, which is to say, we're already making commercial product. So you can trust us that we know how to do that. Where we had to gain their trust was that we could develop their processes for them.
Yep.
So we had to build out that team from having a small technical team to having a real powerhouse for development. So the first 2 years were quite slow. We didn't have a lot of new customers because we were still physically building some of the labs and building this clinical suite with the new technology.
Was the investor comfortable with that, or were they going, Mark, what's going on?
So like any business case, right? You show a plan from A to B.
Yep.
And the way you draw the line of getting from A to B probably isn't what's going to happen.
Right.
So the truth is that after 2 years, we weren't where we wanted to be. It had been slower, just establishing our name, establishing our capability, and really the credibility. So after 2 years, they said, we'd like to see this pick up. And I said, look, trust us, this will. We're starting to get traction now. And true enough, we started to win a lot of business. And our third year was a very strong year of really building out that client base and building the business. And then as we entered our fourth year, there was an opportunity to merge with another company. They were actually slightly larger than we were, but they weren't commercial.
Yep.
So we had something they didn't have.
Yep.
And
you do love a merger, don't you?
Well, the right merger. The right merger. Mergers can be tricky, right? Because there's a lot of things you got to align, not least culture,
Yep.
but vision, you know, making sure that you've got alignment there is really, really crucial.
No, I understand.
So that was actually my preferred path, was to do that merger. You know, Johnson & Johnson were continuing to be a customer. They'd been 100% of our business on day one.
Yep.
They were still similar, actually slightly larger dollar amount of our revenue, but we've been able to bring them down to less than 50% of our business. And my projection was we bring them down to about 30% of our business because we were growing the rest. But 30%, certainly 50% was still a big piece. And there was some nervousness with our investors that, well, what if Johnson & Johnson decide that, you know, they're done and they give us notice? We could be kind of taking a big step back, and now it would take some years to rebuild, fill in what they would leave behind. And so their preference was to explore a sale of the business. So we did that, and we had some strong interest and ended up selling the business to a company called Patheon. Patheon had been a public company. They were taken private under the leadership of Jim Mullen, who previously been the CEO of Biogen, Genzyme's neighbor in Cambridge. And Jim had really done an incredible job of professionalizing and building that business, including a partnership with a company called DSM based in the Netherlands. And he had brought in some of DSM's assets, their contract manufacturing assets, and what they lacked, they had a very strong presence in a number of different areas, but what they lacked was the biologics capability in the US that we had built. So we really fitted in neatly into their overall vision of what they wanted to build. And so we ended up being sold to Patheon. And you know, that was an interesting moment for me, because they wanted me to join and to run their biologics business, which would have been the Genzyme sites. We'd acquired a smaller business based in New Jersey. So St. Louis and New Jersey sites. And then they had a site in the Netherlands and in Australia that had been DSM sites that had become part of Patheon. But, you know, after some soul searching, I realized that I didn't want to go back into a larger organization that had about 10,000 people and 30 sites. That I really, I caught the bug and I wanted to do it again.